Hello guys, today I bring another topic for our upcoming AWES exam, and this is from the chapter Photosynthesis, Absorption and Action Spectrum. Absorption and action spectrum is very important to understand how photosynthesis take place. So absorption spectrum ये बता रहा है कि जब photosynthesis हो रहा है तो plant जो है वो कौन सा light absorb कर रही है और which nanometer of light is absorbed to go for photosynthesis. एंड एक्शन स्पेक्ट्रम हमें बताता है कि किस लाइट पे फोटोसिंथेसिस ज़्यादा हो रहा है. तो जस्ट ट्राई टू अंडरस्टैंड द डिफरेंस बिटवीन देम. लेट्स स्टार्ट द सेशन एंड स्टे ट्यून्ड बिकॉज वी ऑल्सो हैव अ PYQ फॉर दिस टॉपिक. So yes, as I say, absorption spectrum is the absorption of light wavelength of light, absorbed by different pigments. That's called an absorption spectrum. So you can see different pigments are present in the plant, like we have a chlorophyll A, B, carotenoids. These are the different pigments which are actually present in the plant and they absorbs the sunlight. Now if you actually see, a sun has a lot of light, it gives you a lot of different wavelengths, right? But not all wavelengths are absorbed by the plant. Plant absorbed only specific wavelength of light. That is, you can say it can absorb around 400 to 700 nanometer of light. So either it can absorb 400 and range of 400 nanometer to range of 700 nanometer. If you go below this range, the plant will not absorb that light. If you go above 700, plant will also not absorb this light. So the range between 400 and 700 is also called PAR, photosynthetic active region. Okay, so that means between the 400 to 700 most of the photosynthesis takes place. So let's see the graph and try to understand more. Yes, if you can see the graph, so here chlorophyll A is represented by the green, the darker green one, and the chlorophyll A is represented by light green, and carotenoid is represented by yellow color, right? So you can see this is the wavelength of light, 400 to 700. As I already told you, it's called PAR, photosynthetic active region, where most of the photosynthesis takes place between this nanometer only. So if you can actually check, chlorophyll A absorb maximum light at 400 above nanometer, and at around 680 nanometer the chlorophyll A will be more active, right? Then if you check out for chlorophyll B, so chlorophyll B is showing you its activity faster in 450, then there is no activity in between the range, and then it again shows its activity higher around the range of 630, right? So each chlorophyll, each pigment, they show their activity at different wavelength. That means they absorbs this different wavelength of light. And if you actually check carotenoid only absorb around 480 nanometer of light, not any other light, right? So this is called absorption spectrum of photosynthetic pigment. Now next is the action spectrum. Action spectrum simply discuss the rate of photosynthesis. That means at which light or in which light the photosynthesis rate will be higher. In which light the higher photosynthesis take place. That's what it means. So if you actually see, it is indicated by different scientists at different, it is being proved by many scientists, the rate of photosynthesis. Basically it's the main scientist who actually discovered this is Engelmann, T.W. Engelmann, who discovered the actual light in which the photosynthesis is maximum, or the wavelength of light in which the rate of photosynthesis is maximum. So आपको पता है हमारे पास विबगी और कलर है. इस रेंज ऑफ कलर में किस कलर में फोटोसिंथेसिस हायर हो रहा है और क्या नैनोमीटर है उस लाइट का जहाँ पे फोटोसिंथेसिस हायर हो रहा है, दैट इज कॉल्ड एक्शन स्पेक्ट्रम. So we'll see Engelmann experiment also, but first let's see the graph of action spectrum. Yeah, this is the graph of action spectrum. So you can see around 400 nanometer or 450 nanometer, the highest photosynthesis takes place. So 450 represents the blue light. If you can actually say it's Vibgyor, it is representing all the Vibgyor and also representing the wavelength of the light. So you can see in the red light around the photosynthesis rate is higher, and in the blue light the photosynthesis is higher. Why? Because the peak is higher in this range. So we say that in blue and red light the photosynthesis is higher. This is called action spectrum. Now how it is proved? Let's see the experiment of the Engelmann. So this is the experiment what Engelmann do. He take a prism, he pass the white light from there, and he splits the white light into 7 colors as it is being represented, right? Then he take a tank and he grow a specific bacteria which is called cladophora. So the name of this bacteria is this filamentous, this algae, not bacteria. He grow a algae which is called cladophora. So this cladophora algae is grown into a tank, and then he also puts some aerobic bacteria into the tank. And then this cladophora is provided with this spectrum of light, blue light, all the 7 colors were given to this cladophora. So what's happening? He saw that the aerobic bacteria will only accumulate in the region of red light and in the region of blue light. Most of the aerobic bacteria will come to the blue, and most of the aerobic bacteria will come to the red light. So what does this represent? This represents that in blue and red light most of the photosynthesis take place, and that's why most of the oxygen will be there, and therefore most of the aerobic bacteria will be accumulated there. So this is the experiment done by Engelmann to prove that the photosynthesis maximum take place in the blue and red light, and which is shown in a graph, and that is also called action spectrum. Here I have also added some points from a book. You can just take a look over here. You can see the name of the algae is being mentioned in that is cladophora, and also it is showing that blue and red light has the higher range for photosynthesis. So just go ahead and read this paragraph. So here I am giving you a comparison between the absorption and action spectrum. As I told you, absorption spectrum is just showing you the light absorbed by the different pigments, whereas action spectrum will reveal the photosynthetic activity at different wavelengths, or at which wavelength the photosynthetic is high. So let's also check out its graph. If you can see, this is the graph of action and absorption together in one. So here the action spectrum is represented by the black, and the absorption spectrum is represented by blue. So you can see this blue wave is showing or representing chlorophyll A, activity of chlorophyll A, right? So you know chlorophyll A is active at 450 and chlorophyll A is also active at 660. And also the absorption action spectrum is also higher at 450 and 660. That means even photosynthetic rate is also higher at the same range, that is in the blue and red light. So this also represents that most of the photosynthesis activity is done by chlorophyll A, as chlorophyll A is active in that range. So with this we cover the spectrum, both action and absorption. Now we are proceeding with the next part of this chapter, that is light harvesting complex. So if you can actually see, light harvesting complex basically हेल्प करता है सनलाइट को एब्जॉर्ब करने में. तो जो हमारे पिगमेंट्स हैं, जो भी एसेसरी पिगमेंट है, और मेन पिगमेंट तो क्लोरोफिल ए है. एसेसरी पिगमेंट जैसे कि ज़ैंथोफिल है, कैरोटिनॉइड है, दीज आर ऑल एसेसरी पिगमेंट्स जो साइड में रहते हैं, और क्लोरोफिल ए सेंटर में होता है. तो क्लोरोफिल ए हमेशा एक मेन पिगमेंट है, रेस्ट ऑल आर एसेसरी पिगमेंट. सो ये लाइट हार्वेस्टिंग कॉम्प्लेक्स इन सारे पिगमेंट्स को मिला के बनता है, और ये इसको हम टुगेदर कहते हैं फोटो सिस्टम। और ये फोटो सिस्टम हमारे थैलाकोइड मेम्ब्रेन में होता है। टू मेक यू अंडरस्टैंड बेटर, आई एम जस्ट शोइंग यू द डायग्राम इन द नेक्स्ट स्लाइड, सो लेट्स डू द लर्निंग फ्रॉम देयर। So yeah, let's understand the position of the photosystems. So this is what I have drawn is the chloroplast. Inside the chloroplast we have a thylakoid membrane, okay, and over the thylakoid membrane we have these two systems located, photosystem 2 and photosystem 1. So first we have photosystem 2, and then we have photosystem 1, just remember this. Now if I show you one photosystem, how does it look like? Like both the system are same, they have same design. Inside the photosystem only you have all your pigments that absorbs the sunlight. So let's see the diagram of photosystem. In the photosystem you have one of the center part which is called reaction center, in which you will have chlorophyll A. So it's chlorophyll A is in the center, and then it has accessory part attached to it, to which it has all the accessory pigments. For example, it has xanthophyll, carotenoid, chlorophyll B and all. Okay, so these are accessory pigment. Now let me tell you what does these accessory pigment do. So these accessory pigment, they basically take the sun's light, if there is a sun, so they absorbs the sun's light and then they send it to the plant. They send it to the chlorophyll A. All these accessory pigment, they absorbs the sunlight of different nanometer, which is in the range of 400 to 700 nanometer only, and then they send this to chlorophyll A. As you know कि chlorophyll सिर्फ 680 और 400 के रेंज को एब्जॉर्ब करता है. सो अदर रेंज के लाइट को, जो कि 400 से 700 के बीच का रेंज है, वो ये एसेसरी पिगमेंट्स एब्जॉर्ब करते हैं और वो क्लोरोफिल ए को दे देते हैं, जिससे कि क्लोरोफिल ए एक्साइट होता है और फिर ये इलेक्ट्रॉन ट्रांसपोर्ट चेन में जाता है और फिर ये एटीपी बना देता है. दैट इज़ द पर्पस ऑफ लाइट रिएक्शन, राइट. तो करना यही है हमें कि एटीपी बनाना है. बट यहाँ पे फर्स्ट लाइट हार्वेस्टिंग कॉम्प्लेक्स समझ लीजिए. जो एसेसरी पिगमेंट है वो सनलाइट वेवलेंथ को एब्जॉर्ब करते हैं, स्पेसिफिक वेवलेंथ को, और वो क्लोरोफिल ए को वो लाइट ट्रांसफ़र कर देते हैं. दैट इज़ व्हाट फोटो सिस्टम एज. सो ये दोनों फोटो सिस्टम में सेम थिंग है. एक सेंटर में जो क्लोरोफिल ए है उसको हम रिएक्शन सेंटर बोल रहे हैं. लेट मी राइट इट इन द सेंटर. दिस इज कॉल्ड रिएक्शन सेंटर. ओके. और जो साथ में है उनको हम लोग एसेसरी पिगमेंट्स बोल रहे हैं. अब ये जो रिएक्शन सेंटर है यही डिफरेंट होता है PS2 और PS1 में. जो PS2 है उसमें जो रिएक्शन सेंटर है वो P680 है. दैट मीन्स इट गेट एक्टिवेटेड एट 680. सो PS2 में जो हमारा रिएक्शन सेंटर है वो 680 है, और PS1 में जो हमारा रिएक्शन सेंटर है वो 700 नैनोमीटर का है. दैट मीन्स PS1 जो है वो एक्टिवेट होगा 700 नैनोमीटर के रेंज में, PS2 एक्टिवेट होगा 680 नैनोमीटर के रेंज में, जो कि हमारा रिएक्शन सेंटर है. दैट इज़ ओनली द क्लोरोफिल ए इज़ डिफरेंट इन PS2 एंड PS1. बाकी सारे एसेसरी पिगमेंट सेम रहेंगे. सो आई होप यू अंडरस्टैंड दिस. लेट्स हैव अ क्विक लुक. So yes, you can go through these points, and then here you can see PS1 whose reaction center is P700, and PS2 whose reaction center is 680, right? Now PS2 function in the Z scheme. This Z scheme will be the part of our next topic, next class. And they both link through electron transport chain. So definitely this we're gonna discuss in our next session, the Z scheme and the electron transport chain. But for now just understand कि PS1 का जो reaction center है जिसमें chlorophyll A है center में, वो P700 है और PS2 680 है. A better representation of same thing. Photosystem 1 light को absorb कर रहा है, साइड में एसेसरी पिगमेंट है, सेंटर पे क्लोरोफिल है। फोटोसिस्टम 1 में क्लोरोफिल जो है वो 700 नैनोमीटर का है, फोटोसिस्टम 2 में क्लोरोफिल 680 का है। So with this we have done all the topics for today's session. Now I also have some PYQs along with the answers. You can just have a quick recall and revise the thing. So first question is showing maximum absorption chlorophyll A का कहाँ हो रहा है. We know कि वो red और blue में होता है 480 to 600 नैनोमीटर के रेंज में, 660 नैनोमीटर के रेंज में. इंगलमैन ने किस ऑर्गेनिज्म को यूज़ किया था? क्लैडोफोरा को. ओके, फॉर हिज एक्सपेरिमेंट टू प्रूव कि फोटोसिंथेसिस सबसे ज़्यादा ब्लू और रेड लाइट में होता है. LHC लाइट हार्वेस्टिंग कॉम्प्लेक्स, ये ट्रांसफ़र करता है कहाँ पे एनर्जी को? रिएक्शन सेंटर में, जो कि सेंटर पे जहाँ पे आप क्लोरोफिल प्रेजेंट है. ठीक है, P60 एंड 680 एंड P700. विच फोटो सिस्टम वर्क्स फर्स्ट? दिस इज़ अ वेरी इंपॉर्टेंट क्वेश्चन. तो जो फर्स्ट फोटो सिस्टम जो वर्क करती है वो PS2 है एंड देन PS1 है. पिगमेंट्स जो LHC में होते हैं उनको हम एसेसरी पिगमेंट्स कहते हैं, और जो रिएक्शन सेंटर में है वो क्लोरोफिल A है. सो येस, वी हैव कवर्ड ऑल दीज topics. If you still have doubt, you can just go through the video once again, and please like and subscribe the channel and press the bell icon for the notification. For any other doubt or anything that you want me to teach you, you can write it in the comment. Thank you.